The ass worms. Oh my god. After spending hours making, like, I think I'm just gonna start making my stuff to study as I, like, instead of taking normal notes, I'm just gonna start making Quizlets as he's talking.
All right, so here's kind of your resources to think about here and what to expect. So, okay, again, before someone asks, there's not going to be any snail shell statistics questions. That lab was just all about understanding classification, taxonomy, why organisms are named the way they are, and that type of stuff, right? So that was kind of the basis of that lab. No statistics or snail shell classification questions. Our first lab was just more so to get you prepped for all the names that we're going to come at you. So here is lab exam 1 content. We did the protozoans was where our starting point was, Protista. Then we did the Porifera, and then we did the Cnidaria and Ctenophores. We'll get to each one as it comes, and I'll tell you what I'm not going to have on the test. I didn't have any Ctenophores to show you. So we had the flatworms, Platyhelminthes, and we had the pseudocoelomates here. This is just a little discussion thing. If you have questions, maybe someone else can answer. Or you can always message me. Some people have messaged me about what's going to be on the lab exam. Basically anything that was on the PowerPoints, which are available to you there, or that I wrote on the board, which was right here, is fair game. And I did tell you, the types of questions I would be asking. For example, I'm not going to ask you genus names, but sometimes that helps you. Like if you see Plasmodium written on a microscope slide label, even though Plasmodium is a genus name and you don't have to know that name, it would certainly help you know that Plasmodium is malaria, right? So you wouldn't even have to look at the microscope, you just look at the name on the label, and that helps you out. So do you have to know the genus names? No. Does it help? Yes, it certainly does. So, there you go. Okay, so what I'll do is I think we should probably just go through these one at a time. If you don't remember stuff, or if you want to review, we have these little short, some are like 40 minutes, these lab videos that I posted during the 2020 COVID time. These lab videos basically are essentially what I covered in lecture. There is some content on those lab videos that we didn't cover, so you're going to have to filter that. You're going to have to be intelligent and use your in-class resources to make sure that you get the right stuff. Because we were on computers during COVID, I was able to bring some stuff in that I didn't normally have in class to augment it. But just be clear about that. All right. So what to expect here is on. Let me get your. Second sort of process here. Is on the exam. There'll be 30 stations. So we have 4 stations per desk. So there'll only be 4 tables. I know some of the tables have 5 people sitting at it, but you're going to be moving around the classroom the whole time. So there'll be 4 seats per table, like 1 through station 24. 6 times 4 is 24. And then I'll have like an additional 5 stations up here. I think we'll have 29 people up here at that point in time. So there'll be every person to a station. I have a timer going on this computer screen, so there'll be like a 2-minute countdown clock that rotates, and every 2 minutes it'll do like a little school bell, a little ding, and so you move stations. And then I will give you a test page. It's got about it's got 3 pages front and back, so six page faces that will have essentially questions for each one of those stations. So each station will have roughly 3 or 4 questions about it. And the questions will be things like the phylum name, the class name, the feeding structures, if it's a parasite, what the vector or the hosts are, kind of giving you these things along the way of what to expect on these things. Some of them, let's see, habitat, one, I think habitat is a possible option for some of these, considering the parasites need to know the hosts of the habitat. So anyway, let's just go, I'm going to try to give you kind of a hint as to what we're looking for here. Again, you're going to have to know these phylum names, and you're going to have to remember them. Like it's a memorization thing. That's part of the deal. And do I count spelling? Well, yeah. Of course I do. If I said no, I don't count spelling, I would get disaster, right? So, but that being said, if you accidentally, left the T off or something, it's not that big of a deal. Or if you can spell it and I pretty much understand what you meant to write, I probably will let it slide. But if rather than granular reticulosa you wrote grandma, I don't think that one's gonna work for me, right? So just you're just gonna work. There's no hard and fast cutoff for how wrong is it gonna be before you get a green slash, right? So just spelling count, yes. But that being said, slight mistakes will be kind of overlooked for the reason. All right? Sound good? Okay, so along the way, I also kind of indicated to you probably the ones I'm going to really focus in on versus the ones that I'm probably not going to really worry about, right? So there's a whole big list of things here, and I wanted you to see all of them. Obviously, we didn't have any of these, right? So, I can't have on the test if I don't have any, right? So that's okay. And then I think everything else was pretty much fair game there, right? So anyway, realize what we're doing here is, so for chlorophyta, we had 3 different types of chlorophyta, 3 different versions of them. But the main thing was this is chlorophyta. I'm not going to have all 3 of these on the test. I'll probably just pick one, right? Which one do you think I would pick if I had all 3 of these? Which one would be probably the sweet one to pick? Okay. How many say chlorella? How many say chlorella? No chlorella fans out there? How many say cladophora? Okay, some cladophora people. Yeah, so that's pretty good guess. So, all right. So you get the idea. What I'm trying to do though is help you to think about how to study. The best way I think to study is to go, what possible questions could be asked about this, right? So for one,
I looked at these ones in botany too.
Yeah.
Because it's more of the animal life. Probably why you thought that, right?
Full box.
Also, it's pretty easy to see, right? And then, okay, that's one question. What is the phylum name of this organism? Blank. This organism is in the phylum blank. You have to write chlorophyta. What else could I ask about it? What's that? What other questions could we ask about that little lowly bulb icon? Yeah, or maybe what kind of organization is it, or I could say something like it's a cluster of cells called a blank, So something that asks you about what's a colonial organism, right? It's a colonial thing.
And chlorella.
What other questions could I
Chlorella
ask?
is unicellular.
How does it move? How does it move?
Is multicellular.
Yeah.
What does it use to move?
Flagellum.
Flagellum? It has flagella?
Yeah.
So each little cell has its own flagella, and it kind of uses that to move. Cool. What other questions could I ask you about this? How about what other kingdom is most closely related, right? Like this is the most closely related to what other major kingdom of life? What's that? Plantae, yeah. So this is basically the bulb boxes, or the bulb box. The chlorophyta is kind of like the food plants, it's like the ancestors or close relatives of the plants, right? That was the significance about it, was it had some animal-like traits, it has some plant-like traits, but basically the chlorophyta is its own thing, right? So it's there. So the purpose of that little exercise was to get you thinking about how you might be able to study test is to go through the images and kind of think about, okay, what was the important things that were mentioned about this thing, about these things. Okay, retorta monada. Let's see here. I had some Giardia on a slide. I mentioned to you that the Giardia I had, didn't look very good. They kind of look like little broken up footballs, like pop footballs or something. So it'd be really hard for you to actually recognize that as Giardia if I didn't have other hints available. So this would be a case where having the label exposed on the slide that said Giardia, nameless, would be really helpful. So just a case there. I can't remember. I think last year I didn't have Giardia on the test. Because I have other groups or whatever. But it depends. I mean, this is a big class. So sometimes with a big class I have to kind of scrape for extra questions. But if I had Giardia on the test, let's say I'd ask you the phylum, right? I might ask locomotion, right? That's about it. I typically don't have. Well, I could ask you how you get the parasite, right? How you acquire this infection. How you drink, untreated water, right? There you go. That's pretty good. But this one I haven't always had it on test. This one I usually do though because it's kind of interesting. Gift that keeps on giving. Phylum Axostylata, so phylum name's important. And the fact that you get the parasite by sexual transmission, that's basically close contact, or
Contaminated.
sharing underwear, things like that. And I have flagellum, and then there's a structure inside. It's kind of tricky because the phylum name is Axostylata, and then the structure inside is called an axostyle, right? So it's kind of like a similar answer. But I think I told you what the axostyle probably is useful for. The actual answer is not known, but most likely, what is the axostyle? What does it do? Yeah. Yeah, it's like a flagellum anchor or like a flagellum support. So be specific. Like I know in years past, some people have written locomotion. I'm like, all right. That's kind of hard. Like it's not quite right, and it's not totally wrong, but but the better answer would be it was basically the flagellum is attached to the base of the axostyle. So it's probably some sort of a support for the flagellum when they're making their way to their destination. Okay. Euglena, I put these on test sometimes. The problem with euglenas, they're so small that it's kind of hard to really identify, because they're so tiny. And all the ones that I have, of course, aren't stained green, which is the interesting euglena. Instead, they're usually purple or something. And also, it's hard for me to leave the cover slip slide label uncovered, because the name Euglena is so much of the actual phylum name, it's just kind of confusing, right? So anyway, maybe not. But if I did have Euglena on the test, which I have in the past, what would be a good question other than what's the phylum name, Euglenozoa, what's its locomotion, flagellum? What's the other interesting thing about the Euglena that makes it interesting?
The eye spot. He had a different
What would be a chlorophyll? Yeah, so it feeds by sunlight energy, by chlorophyll, when light's available, but it also, feeds by using its cytosol, its little cell mount, if there's, organic material available. So, I mentioned that the chlorophyll, the chloroplasts, are kind of transient, like they can lose their chloroplasts and then just feed like a paramecium does, or they can reacquire chloroplasts. It's kind of an interesting thing. That'd be a good one. But they are kind of hard because they're so small, it's hard to really tell what they are like in that 2-minute time period. I do think about that. I think when I take the test, I'm not trying to trick you or anything at all. Although there are some good questions I'll tell you about. Not this test, but down the road. Okay, more likely though, I like this one. Because this one, I can leave the label uncovered, trypanosoma, but I can still ask you the phylum name, right? It's the same phylum name, Euglenozoa. And then in the trypanosoma, I can ask you what the, depending on where it's found, what the disease name is, right? And I think I told you which one I probably would use, right? Because it's the one that probably killed Darwin, right? Darwin probably died from what disease? Chagas disease, right? So Chagas disease is the Central American sleeping sickness, and it's transmitted by kissing bug. Kissing bug is the vector for this. Got it? These are all kind of fair questions. Vectors, kissing bug, Chagas disease, Central America, killed Darwin. That's the one I like. So here's the thing: if the question says, in Central America, what is this disease called? Don't say African disease. That's not serious, right? So it's Chagas disease. All right, there you go. Certainly malaria will be on there because up until HIV took over in the 80s and 90s as the leading cause of pathological death in the world, malaria was the primary pathological cause of death. Malaria, cholera, and then HIV took over. But anyway, so what questions could I ask about this one? The phylum, Aphicomplexus. Because I could leave the little slide labels with Plasmodium exposed so you know it's malaria. And then Aphicomplexus is a phylum. I could ask you what the vector is. It's a mosquito, right? I could also ask you what the mode of locomotion is, right? What the locomotion mechanism is. And what is it?
Mosquito. Vector.
What's that?
The vector.
So what's the mechanism for locomotion? They don't have one, or the vector. The vector is how they get around. And then, but otherwise, they don't have any flagella or cilia or anything, so there's no locomotion. So either way, it'd be fine. Okay? Any other questions on that one? That's pretty good. That's 3 questions. So you can see, like, 3 or 4 questions for each little thing to be good. Certainly, plasma and volume on there.
I just don't.
I don't have any toxoplasma. That's a fun one. Ciliophora, guaranteed to see Centaur on there, right? Because Centaur is my favorite. So anyway, so that is a Ciliophora. Yeah, what other questions? They move by cilia, which I know is a redundant question. Cilia is a locomotion mechanism. And I'm trying to think what other. What's the structure called where these organisms pull food into their cell? What's that? Cytostome, right? Cytostome, which literally just means cell mouth. So that would be a good question for that. Like, they collect food and ingest it through the blank. Cytostome is the correct answer.
I think I know.
Got it? Okay, cool. Probably won't have serratium on the test, but I will for sure have a chunk of coral, right? A chunk of coral, because the chunk of coral is a combination of a dinoflagellate and what other group? So an anthozoa polyp, right? So the coral is a symbiosis between dinoflagellate, Zooxanthellae genus, and then inside an anthozoan, Cnidaria polyps. And that's what the fusion of the two is called coral. Right? So probably Dinoflagellata, the answer will come up with a chunk of coral, not with serratium. Oh, I like serratium. It's kind of cool. So we'll see that one again. Amoebozoa, sometimes I have those on a test, but actually better off would be to use one of these. The granula reticulosa has the same locomotion mechanism as the amoeba, right? And what's the locomotion? They feed and move by using what? They don't have feet. They don't get feet until arthropods. What do they have?
Fake feet, right?
What? Pseudopodia. Pseudopodia, which literally means fake foot. So I know what it means sarcastically. Pseudopodia is the correct answer, right? If you wrote foot, I'd have a hard time giving you the right answer. If you wrote fake foot, I might consider it, but depending. If you just forgot the word pseudopodia. So anyway, the granular tribulosa basically has the same kind of questions that the amoeba would have, but I got a couple more questions I could ask, like what is the shell or test made of?
CaCO3.
Calcium carbonate, right? So it's calcium carbonate or CaCO3 or calciferous. I think all three of those answers are okay. Calciferous, calcium carbonate, CaCO3, those are all good answers. Okay.
If you just say calcium.
Anything else on this one? You good? This is a good one. I could also ask radiolaria. And the main difference here is it also feeds and moves by pseudopodia, but its shell is made of what? Silica. So it has this silica or SiO2 is the shell material. Okay, I don't have clinoflagellates, so that one can't be on there, but I do have sponges, so yeah. All right, so on to the sponge lab. SpongeBob. Okay, so we have a couple important things for sponges. The main thing is we have organization of cell material. So this organism has no true tissues. So if I have like a commercial sponge at a station, and I ask you, what's the orientation of this organism? It has no orientation. It has no symmetry, right? So it's asymmetrical or no symmetry. Also about the tissue layers, there's no true tissues. So, an answer can be none if this is sponges, for example. And so for the commercial sponge, the big one, I'd probably ask you the phylum name. I'd probably ask you the class name for the commercial sponge. I'd probably ask you about symmetry, tissue layers, those types of things. For the slide mount specimens of the Grantia, certainly I'd be asking you about choanocytes. Choanocytes are the important feeding structures of the sponge. So you might see a slide that looks something like this, zoomed in, and pointing towards those little cells on the inside. And oftentimes I'll give you a little bit of a hint as to what it is, because I understand sometimes when you look in the microscope slide and you see just that, even I, if someone were to blindfold me and put me in front of a microscope and then just show me the top image without the context, I probably wouldn't know what that was. I'd make a good guess, but that could probably just as easily be epithelium of human skin or something. But so oftentimes on a test question, I'll give you a little hint, like this sponge cross section. The pointer is identifying a blank cell, a unique cell of the phylum. So I'm trying to give you a hint. So in a lot of cases, if you read the question very carefully, there's like little hints in the question that will lead you to the right answer. Make sense? So I would say like a good 20% of the answers on this test, you could answer without even looking at the specimen, without reading the question, right? So just as a hint. That being said, I think it's really important to actually read the whole question before you start answering stuff, because sometimes you'll get cued in along the way. Okay, so the key types here is this is pointing at choanocytes on the inside of the feeding cell. There's also these little blobby ones here are called amoebocytes. Those are the food transport cells. I don't have any examples of pinacocytes, so I mentioned they're too thin to show. We'll point out. But the choanocytes and amoebocytes are good to put on one slide. Also, this cavity here in the middle is called what? Spongiocele, right? Sponge cavity is called the spongiocele. And so that's kind of cool. And then the opening at the top of the sponge, the opening at the top where the water exits the body, what's that called? What's that? Osculum. Osculum is the vent where the water exits the sponge's seal. So this is a diagram showing this. That water is drawn in the ostia. The ostia are so small that I don't really have any way to show them on exam because they're too small to look at. But the osculum, you can certainly see that in the Lucasolinia slide. So that little green Lucasolinia looks like little fingers. At the very, very top, there's a little hole that was where the osculum was. That's a good one to ask. So anyway, and then the Lucasolinia also is in the class Calcarea because why? What makes it in the class Calcarea?
How do you know that the spicules are made?
What's that? Spicules? Spicules are made of what? Calcium carbonate, right there. Right. The calcium carbonate spicules is what is the defining feature of the class Calcarea. Now you don't know their calcium carbonate unless you test them, but you should just know that Lucasolinia is in the class Calcarea. So if I have the Lucasolinia on a test, you're going to have a Lucasolinia on a test. Anyway, questions I could ask are: is it in the class Calcarea? I could ask you why that is, or what about it makes it class Calcarea. That's calcium carbonate spicules, or calciferous spicules. And I could ask you about the little hole at the top of the sponge body, and that's the osculum. Got it? Sound good? Okay, Hexaconellata, I have one specimen. Probably I'll have it on there. And again, it's the class name, Hexaconellata. It has silica spicules. And on this one is where I might ask you a question about symmetry. Like this organism shows what kind of symmetry? Zero symmetry, right? No symmetry. Or asymmetrical. Anyway, so that's pretty straightforward. Only a couple questions on that one. And then the commercial sponge is the class Desmospongia. And the key feature of this one is it has sponge fibers that are these elastic protein fibers that support the body. Got it? Okay, sounds good. Desmospongia. Certainly I'll have a commercial sponge on there at some point. Maybe right here. All right, that's it for sponges. Questions about that? So, okay, along the way, I'm just trying to give you hints as to what specimens will be on here, what kinds of questions will be asked. So it's your best interest to write them down or record this stuff or, whatever. Get together and ask questions about those things. Here's some terms you might need to write on a test, Again, pinacocytes, I don't have any examples to show you, but choanocytes, aminocytes, spicules, spongoceal, osculum, spongin, those are probably answers you might need for some questions. Okay, let's go ahead then and look at the Nigeria. I did want to point out that some people were kind of thinking, when we had our Nigeria lab, there's only a few things to look at, no big deal. But realize when you put all these labs together, it's like, wow, it's a lot of information. Right? So it starts becoming overwhelming when you put it all together. So the phylum Nigeria is the main one. I'm not going to have any ctenophores for this. I don't have any specimens to show you. And then, so the key thing with these is they had this diphloblastic development. They have endoderm and ectoderm, so tissue layers. So they have two true tissues. So there will be a question somewhere on this test asking you how many true tissue layers does Nigeria have?
Tissue layers.
Or endoderm and ectoderm.
And
The symmetry. They have a radial symmetry, kind of a circle-shaped symmetry. And then the unique feeding cells for these is the cnidocytes, feeding cells. And so that's kind of the main ideas. Also, life cycle, the medusa versus the polyp stage. all kinds of information here. Okay, so let's go ahead and take a look at, certainly I will have a cross-section of a hydra somewhere that has a pointer pointing towards the stinging cell called the cnidocyte. So remember I said that these cnidocytes basically are a stinging cell that's unique to this group. So probably I'll have a microscope slide that shows a cross-section through the hydra that shows cnidocyte, but also a couple other things could be asked, like the gut cavity is called a gastrovascular cavity. If they have that gut pocket, it's called a gastrovascular cavity. It's lined with endoderm. The jelly layer in a jellyfish or the hydra is called the mesoglia layer, right? It's kind of proteinaceous layer between the ectoderm and endoderm. So those are all good questions for the hydra cross-section. Especially the cnidocyte. Okay. Good. So for the hydrozoa, again, the cross-section through the hydra arm is good. I also have that floating manowar thing, and so probably we'll have a manowar on the test. So what kind of questions could I ask on this? Not only is it a class hydrozoa, what was kind of an interesting thing about this that was totally weird? What's that? They're colonial? Good. They're colonial, and also, what else about them being colonial? It's actually a floating polyp. It's not a medusa stage, right? So it looks like a jellyfish, but it's actually a floating polyp. And those are good answers there. Okay? And if I had Ophelia on the test, these are also polyps, but there's two different types. There's feeding polyps. The ones with the tentacles are for feeding. And then what happens is they mature and they become these structures. What were they called? What was this structure called? Kind of looks like a cone, pine cone. It actually has the same name as a angiosperm male flowering cone. Strobili. Yeah, strobili. Strobili is, that's the same name you use for a male pollen cone plants. But strobili is when the feeding polyps mature, the strobili, what's the function of strobili? What are they making?
Babies.
Yeah, they're like Medusa dispensers, right? So each one of these little discs in here is a baby Medusa that will pop out and swim away. Isn't that cute? So anyway, so here's some Medusa emerging, but the strobili are the asexual Medusa producing structures on the polyps. Okay? For Medusa, I'll probably have a moon jelly. So Scyphozoa, because it's the big jellyfish in the dish. So you see a jellyfish in the dish. A couple good questions would be, what's the class name? Scyphozoa, the true jellyfish. What life stage is this? It's a Medusa stage, right? What else? Oh, what kind of symmetry does this organism show? Radial symmetry. Here's a additional one that I ask sometimes people forget, but I think I mentioned it pretty clearly. What are these little horseshoe-shaped structures in the middle? Gonads, right? Okay, good. Gonads, right? So, so the horseshoe-shaped structures are gonads, and so that's a fair question on that one. So I did like 4 things for the blue jelly in the dish. I'm not going to have any of the other life stages slides. Okay, so just the blue jelly. I mean, I made you look at those because I wanted you to appreciate the idea of this alternation of polyp to Medusa stage. But here I'll have the Medusa stage for Aurelia, and then we'll have the Lobelia polyp as your polyp stage, right? As well as the Mycelium. Okay, I don't have any of these, right? But I want to show them because that was interesting. information. Yeah, I do have these anemones in the dish, and those are pretty fun to put on there because a couple things about them. Class Anthozoa. The anemones have a folded gastrovascular cavity. The cavity is kind of like folded, so it has more fold to it. And there's no Medusa stage. It's all polygonal. There is no Nigerian that's Medusa only. They're either polyp Medusa or just polyp. So, also, I think with class Anthozoa, as I mentioned before, this image again, we saw this before, that the coral is symbiosis between the phylum Nigeria class Anthozoa and the phylum Dinoflagellata in the protists. So you probably should be aware of that. So I'll have one station that might ask you about both of them together. Got it? Okay, and that was it. No ctenophores in the test, so we're good there. All right, Platyhelminthes lab. So this is a good one, the phylum Platyhelminthes. And a couple key things here is this is triploblastic development. They have ectoderm, endoderm, and a new tissue layer called mesoderm. They have bilateral symmetry, and they are a combination. There's some parasitic lifestyles in here that you'll have to know some parasite history on these. But the ones I do have are the class Tribularia, Trematoda, and Cestota. I don't have them on video to show you.
No.
That's right. What question? Okay, so certainly, we'll have some flatworms on the test. Probably a cross section. And if I were to put a cross section on the test, which one do you think it would be?
The middle one.
The middle one, right? This has got the most interesting stuff in it, right? The middle cross section has this pharyngeal cavity, pharynx cavity, and it has the pharynx inside, and then it has the gastrovascular cavity on the sides. So there's a lot I can ask. On the anterior and posterior one, not a lot to ask, so I probably won't have those. So, good thing to ask here would be what tissue layer is the gastrovascular cavity aligned with? And that's endoderm, right? Ectoderm on the outside. Interestingly enough, is the lining of the pharyngeal cavity is also ectoderm. So this is an ectodermal lining in this cavity here. Basically, it's outside the organ. Folded side. And then the mesoderm is this kind of connective tissue and also the surrounding. Okay? So plan on a cross-section of the pen area, probably the pharyngeal cross-section. Don't know if I'm going to have I can't remember if I have a whole mount one. But I think it's a lot easier just to use like a clone or something. So probably more so to have one of these to ask you about the bilateral symmetry, or maybe the tape one would be a good one for bilateral symmetry. Okay, so for the parasites, we had the Trematoda are all parasites. What is the intermediate host of all the Trematodes?
Snails.
Snail. So an aquatic snail is always the intermediate host of every one of these species, parasite life cycles. So there'll probably be a couple questions about that at different stations. But an aquatic snail is always going to be a host along the way. Okay, so the human liver fluke. How do you get this one? How do you get this disease? Yeah, so raw or undercooked fish. Perfect. That'd be a great answer. Okay. And it has a snail host. Okay. As far as what to look at here, the important things would be like it has an oral and a ventral sucker, right? Oral or central sucker. Nice, central sucker. There's the gastrovascular cavity on the side. You know, I don't think I'm gonna ask about the gonads on this one, but I wanted to just point those out. They're pretty conspicuous. Okay, and then the fasciola. Oh, the fasciola it's kind of hard to put the whole worm on a microscope because they're so big. But I'll probably have one of the sub-life stages. Which life stage do you think I'd put on there? As so, like, you know, with the Clonorchus, I can fit that onto a microscope slide that you can answer questions on for it being a fluke, to be adult. But which of these stages do you think would be like the easy one or the one that would be the most interesting for the test? The what? What's it called? Cercaria. So probably, if I had a fasciola life stage on a microscope slide, it'll probably be the cercaria stage. Okay. And because there's also a couple of things I could ask about that. What did the cercaria just emerge from? Like, where does the cercaria come from? Always comes from what? What? Snail. Yeah, so the snail host is what produces the cercaria. So that's kind of interesting.
Snail actually produces,
and that came up again in this one. The swimmer's itch or the schistosomiasis is here's a snail here and here's all these cercaria floating up out of the snail. So the cercaria are the stage that then drills into the next host. In the case of schistosomiasis, it drills into your skin, or in the case of swimmer's itch, it drills into your skin and dies there, causes a rash. But the cercaria stage is the infective stage, right? So probably in fasciola, I'll have a little. They look kind of like this, a little cercaria slide, and I'll ask you probably where this parasite stage just came from and where it's going to. Right? In the case of fasciola, it drills into a plant, and then the sheep or cow or human or whatever eats the plant, which gets the parasite that way. Okay. Finally, certainly I'll have tapeworm on the test. Maybe even 2 stations of tapeworm. They're so interesting. Guaranteed there's going to be a head end of a tapeworm on there. So what are some key things you're going to find on the anterior or head end of a tapeworm? Like I can say, this organism is in the class blank, Cestota, cancer. And then the pointer is aiming at the, what is that structure? Scolex, right? Scolex is the head segment of the tapeworm. And then what are these segments just following the head called? Proglottids, and the ones that are the most anterior contain mostly what? What kind of gonads?
Female gonads.
Female is an trickle later. So the male gonads here first. Testes are in the what are called immature proglottids, right? So listen up. I think I just told you something in that lab. Is that there's a scolex and there's segments right behind it. You know, these don't have anything in them, but as they go back, they gradually begin to form testes. So the immature proglottids are basically male, mostly. Then, as you go farther back, you can see here, here's the head. You go farther back, maybe about a third of the way down the worm, and you begin to see female gonads forming. So the proglottids will have both male and female together. And then as you go further back, about 2/3 down the worm, the male parts are gone, and it's only female parts, right? And then at the very, very end of the worm, the right proglottids have fertilized eggs in them, right? So that's kind of the progression, is the proglottids are formed at the scolex, and essentially that pushes back these segments that as they get older, they go from male to female to eggs, right? Sound good? So, if you see a skull X, you know it's probably going to be the male structures by the skull X. And if you just see proglottids with eggs in them, you know it's the right ones at the very end, female stuff, right? Female structures. Questions? Probably won't have one from the middle that has both male and female, and I ask you to identify anything that's kind of too difficult, right? It's like, well, challenging for the mature proglottids to know, is it male or female, or is it both, right? Because it's kind of transitional. So I'll be very clear. Got it? Okay. Okay, and then how does your dog get tapeworm? What's that? Nope. Not eating poop.
Mouthing another dog?
No, that's nematodes.
Oh. Oh, the flea.
Eating fleas. It ingests the flea. That the flea has eaten, the flea ate poop, the eggs off the poop, the flea larvae, and then the flea adult, when it gets on the dog, is ingested by the dog. And that's how the dog gets tapeworm. Gotcha? Okay. And I won't have any questions about pork tapeworm. It'll be all big dog tapeworm. Okay, we looking good? Is this helpful at all? All right, just telling you what's on the test. All right, here's the last lab. It is, the group generally is called the pseudocoelomates, which is not really a true taxonomic name, but it indicates kind of a body plan, right? So somewhere along the way, there'll be a question about pseudocoelom, right? Probably maybe one or more questions. We have 3 phyla. I have all 3, so expect probably all 3 of them on the test. So probably I will have, well, let's see here. For, the easy thing about these was they were all found in nematoda. It's just I had different parasites, right? So the Ascaris, if I had the Ascaris on the test, which is this one, here's the Ascaris.
Oh my god.
What types of questions would I ask? I did not have examples of male and females for you, so don't worry about that. Yeah, I won't ask you genders. But like, for example, how do you get Ascaris as a parasite? Yeah, ingesting eggs, usually from unwashed manure fertilized bird vegetables, right? Good. And then I probably will have a cross section of the test. I think all you saw that. I could ask all kinds of cool things on here. Namely, the cavity here is called the pseudocoelom. And the main reason it's called a pseudocoelom is because the outside is lined with mesoderm, but the intestinal tract is entirely made of what? Endoderm, right? So this intestinal tract, gastrointestinal tract, is entirely made of endoderm. Right? And that's what makes this a pseudocoelom. That one's pretty obvious. I'm not going to ask you about the gonads on that one. So it'll probably be endodermal gut, pseudocoelom, and then a mesoderm lining the outside of the pseudocoelom. Trying to get the idea you understand what a pseudocoelom is, right? Very good. So plan on a cross section of an Ascaris. It'll look something like that. Probably have a point directed gut. Maybe one of the numbers on the number scale, identifying the cavity. There you go. Got it? These are all available online too. Probably we'll have a hookworm on the test. Now, they were so small you couldn't see the hooks of hookworm, right? So it was nice that the slide label says Decatur on it, or the one, the vial said ankylostoma, probably Decatur we'll have on there. And then how do you get hookworm? Yeah, barefoot, tip to the tulips, right? You walk in pastures barefoot where animals have pooped, including cattle or dogs or people, and that's how they drill through your foot, right? Fun. All right. And probably we'll have pinworms on the test because they are the most common nematode parasite in humans, even in America. So, pinworms, how do you get them? Here you go. Scratch your thumb, suck your thumb. Right. Okay. And any questions about that? All right. And then trichinosis. How do you get trichinosis?
Wild bear.
Yeah,
Or
uncooked
wild
bear or
pig.
pig meat, or rat meat, that you think. Or human meat. With box beans and canned beef. Okay, anything else there? Trichinosis, so probably have trichinosis on the chest. I like it because it's got the little worms embedded in the muscle. And how you get it is undercooked pig or bear meat usually is where it comes from. Okay? Filarial worms, the only one I have is the dog heartworm, right? So that one, you should know that that part in the jar is dog heartworm. How does your dog get heartworm?
Mosquitoes.
Mosquito vector. So it's a trio mosquito specifically, but mosquito's good enough. Okay, so mosquito vector for the dog heartworm, that's a good one. Maybe that's station right there. Okay, and then I'll probably have one of our nematomorpha specimens.
Of course.
You know, and good couple questions on this one. The hosts are all insects of some type, so that they're entirely, the whole group is parasites of insects. And then I will probably also ask you, what is the common name from folklore for this group? And you can either write horsehair worm or Gordian worm. Either are correct. I think it's interesting to know though. And then a follow-up question would be, why is this group separate from Nematoda? What makes it different?
It only affects insects.
So the Nematoda, they look like nematodes, but they're not nematodes. They're closely related, but they're not nematodes. Why?
I think it's their guts.
Yeah, there's no complete gut tract. So these have no gastrointestinal tract. They probably lost it along the way. They absorb their nutrients from the outside. So they are separated from the nematode because they lack a gastrointestinal tract. Got it?
The last day before.
And we'll probably have a rotifer on the test. And this is also has a pseudocoelom, one question. But then the locomotion and feeding is done by, what's that structure at the head end? Ciliated corona, right? It's got the ciliated, what's called a corona, which means crown. But it has two ciliated discs that it used to move around and feed. All right? Good. All right, so I think that summarizes kind of the questions that'll be on here. If you want to look at these specimens, you're welcome to.